Hello students, I am Dr. Tisha Vyas. Welcome all of you to our online learning session of biotechnology. It's been my indeed pleasure and a matter of great happiness that I am interacting with you after almost 3 months, isn't it? I hope even you are eager and excited to start learning for the new term. So today we are going to have a brief introduction of what biotech subject is and its history that led to an innovative technology in biology, that is biotechnology. The content of the Unit 1 includes introduction of biotechnology, the historical background of biotechnology, what does it mean? That is the definition of biotechnology, its contributions from related disciplines, the major areas and applications of biotechnology, impacts of biotechnology on national and global economies, as well as the challenges and the future of biotechnology. So let us have a look at the definition of biotechnology. Biotechnology is a broad area of biology involving the use of living systems and organisms to develop or make products. Depending on the tools and applications, it often overlaps with related scientific fields. In the late 20th and the early 21st centuries, biotechnology has expanded to include new and diverse sciences such as genomics, recombinant gene techniques, applied immunology, and development of pharmaceutical therapies and diagnostic tests. If we have a look at the wide concept of biotech or biotechnology, it encompasses a wide range of procedures for modifying living organisms according to human purposes, going back to domestication of animals, cultivation of the plants, and improvements to this through breeding programs that employs artificial selection and hybridization. Modern usage also includes genetic engineering as well as cell and tissue culture technologies. The American Chemical Society defines biotechnology as the application of biological organisms, systems, or processes by various industries to learning about the sciences of life and improvement of the value of materials and organisms such as pharmaceuticals, crops, and livestock. Biotechnology is the application of scientific and engineering principles of the processing of materials by biological agents to provide goods and services. Biotechnology is the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. From its inception, biotechnology has maintained a very close relationship with society. Although now often associated with the development of drugs, historically biotechnology has been associated with food, addressing such issues such as malnutrition and famine. The history of biotechnology begins with zymotechnology. Zymotechnology is the old term used for the study of the processes of fermentation in yeast and bacteria that are used in the production of foods and beverages such as bread, cheese, tofu, beer, wine, steak, natto, etc. Of course, these practices go back to the ancient times. However, in the 19th century, with the rise of big industries, particularly in Britain and Germany, technoscientists began to isolate the microorganisms involved and to study them. With the techniques of scientific biology of the 19th century, it becomes possible to isolate pure strains of various yeasts and molds involved in these processes. so as to standardize the mass production of these products. In the regards, at the end of the 19th century, various industrial and governmental labs and teaching institutions were established. By World War I, however, zymatechnology would expand to tackle larger industrial issues, and the potential of industrial fermentation gave rise to biotechnology. The Office of Technology Assessment of the U.S. Congress defines biotechnology as any technique that uses living organisms or their products to make or modify a product, to improve plants or animals, or to develop microorganisms for specific uses. Let us have a look at the history behind biotechnology. There is a common misconception that biotechnology is relatively new because it only involves working with DNA and genetic engineering. But biotechnology is not new. Humans have been manipulating living things to solve problems and improve their way of life for millennia. The origin of biotechnology date back nearly 10,000 years ago to early agrarian societies in which people collected seed of plants with the most desirable traits for planting the next year. There is evidence that Babylonian, Egyptians, and Romans used this same selective breeding practices for improving livestock. As far back as 6000 BC, people produced beer, wine, and bread using fermentation, a natural process in which the biological activity of one-celled organism plays a critical role. By 4000 BC, the Chinese were using lactic acid producing bacteria for making yogurt, moles for making cheese, and acetic acid bacteria for making wine and vinegar. By 1500 AD, plant collecting expeditions became quite common across the globe. The collections led to the establishment of the first plant gene bank. Plants with desirable traits, including resistance to diseases, were stored for future breeding purposes. A Russian geneticist and agronomist initiated a comprehensive research and breeding program that included a logical plan for crop genetic resource management. The end of the 19th century was a milestone in biology. Microorganisms were discovered as well as pasteurization. Gregor Mendel began the study of genetics using seeds and plant experiments. The term biotechnology was coined in 1919 by Karl Erecki, a Hungarian engineer. At that time, the term meant all lines of work by which products are produced from raw materials with the aid of living organisms. Biotechnology brought industry and agriculture together at the beginning of the 20th century. Fermentation processes were developed that produced acetone from starch and paint solvents to be used as automobiles. Between 1930 and 1952, researchers focused their effort on the relationship of genes and proteins. They found a direct relationship between mutations of a gene and the sequence of amino acids in a protein. The discovery of penicillin during World War II led to the focus of pharmaceuticals. The Cold War years were dominated by work with microorganisms in preparation for possible biological warfare and the production of more antibiotics. The final threshold to modern molecular biology was crossed in 1953 with the discovery of the structure of DNA by James Watson and Francis Crick. Experiments by many other scientists soon followed to determine how the information in the gene is decoded and expressed. The accumulated knowledge of the cell structure, biochemistry, and heredity opened the door to modern molecular biology and biotechnology. In the year 1985 is the Human Genome Initiative, and its goal is to discover all of the 80,000 to 1 lakh human genes by the year 2003. And nowadays, biotechnology has achieved its greatest advancement in the field of recombinant DNA technology, PCR, cell structure, fusion, gene cloning, DNA fingerprinting, environmental engineering, immunology, etc. Let us have a look at the important events that took place in the past and that lead to the development of biotechnology. 6000 BC was the year of Sumerians and Babylonians that knew the technique of preparing beer. 2500 BC, Aryans started preparing curd. 2000 BC, Greeks were probably first to prepare cheese from milk. Cheese, one of the first fermented food. 1822 to 1895, Louis Pasteur, the father of microbiology and the father of industrial biotechnology, discovered that yeast produced alcohol in wine and rod-shaped bacteria produce acetic acid, causing souring of wine. 1929, Alexander Fleming discovered antibiotic penicillin. 1973, first successful genetic engineering experiments were carried out in famous world laboratories. This particular image is of the biotechnology timeline, which also tells you about the different events that took place and that lead to the revolution of the biotechnology subject. The very first event took place in 8000 BC, that is, biotechnology begins as human begins choosing or altering plants and livestock so they can be domesticated. Potatoes becomes the first cultivated food. In 4000 BC, the Egyptians mastered the art of wine making. Further, 2000 BC, Egyptians and Sumerians learned brewing and cheese making. In 500 BC, in China, moldy soybean curd becomes the first antibiotic to treat infections and ailments. In 300 BC, Greeks developed grafting techniques for plant breeding. In 1950, Dutch spectacle marker Zacharias Janssen invents the microscope. 1663, English physicist, mathematician, and inventor Robert Hooke discovers the existence of the cell. 1675, Dutch students of natural history and microscope maker Antoinette van Leeuwenhoek discovers bacteria. In 1796, first smallpox vaccine is discovered. Edward Jenner discovered the process of vaccination by inoculating a small boy with cowpox and then trying to reinfect him with smallpox. The boy recovered from the weaker cowpox infection and thus became immune to smallpox. The cowpox virus was called vaccinia from the Latin word for cow, vacca. This is how the word vaccine came into existence. 1833 was the year for the first enzyme discovered and being isolated. 1838, Swedish chemist Jöns Jacob discovers proteins. In the year 1839 to 1855, German scientist proposed that all organisms are composed of cells. 1855, the E. coli bacteria is discovered and it later becomes a major research, development and production tool for biotechnology. In the year 1859, Charles Darwin's landmark book, The Origin of Species, is published. 1861 is the year for the French chemist Louis Pasteur that develops pasteurization, which is a process that protects food by heating it to kill dangerous microbes. 1885, vaccine for rabies disease discovered, and Pasteur vaccinated a young boy who had been bitten by a rabid dog. This vaccine was made from the extract of the spinal column of a rabies-infected rabbit. A modified version of this vaccination is still used today and has saved thousands of lives. 1865. After 7 years of cultivating and testing thousands of pea plants, Gregor Mendel publishes a description of rules governing how heredity traits passes between generations, which became the foundation of modern genetics. In the year 1919, the word biotechnology is used in print for the first time. The year 1870 to 1910, father of modern plant breeding, Luther Burbank develops over 800 new strains of fruits, vegetables, and flowers. His blight-resistant Burbank potato is heavily planted across Ireland, ending the potato famine. 1922. In Toronto, Dr. Frederick Banting and his assistant Charles Bett discover insulin as a treatment for diabetes. The year 1928, Scottish bacteriologist Sir Alexander Fleming discovers penicillin as an antibiotic. 1941, Danish microbiologist Justin coins the term genetic engineering, a technique that is used to transfer selected genetic material from one organism to another. In the year 1942, there was careful breeding cantaloupe mold in large tanks. American microbiologist Andrew Moore develops a technique of producing penicillin in large quantities, launching its career as a wonder drug. 1943, Canadian scientist Oswald Theodore, a very isolated pure DNA. 1953, James Watson and Francis Crick are the first to describe the double helix structure of DNA. 1958, DNA is produced in a test tube for the first time. In the year 1961, mRNA was identified for the very first time. The year 1962 was a very remarkable year as scientists Francis and Crick won the Nobel Prize for the discovery of double helix structure of DNA. 1968, scientist Marshall and Hargobind Khurana wins the Nobel Prize for the genetic code of the 20 amino acids being identified. 1970, Norman Borlaug becomes the first plant breeder to win a Nobel Prize for his work on new wheat varieties that increases yields by 70%. 1971, the first complete synthesis of a gene. The year 1973 is known for Stanley Cohen and Herbert Boyer development of recombinant DNA technology. 1976, The sequence of nucleic acid base pairs that combines to make DNA is determined for the first time for a specific gene. 1982, the first recombinant DNA vaccine for livestock is developed. 1977, Herbert Boyer, founder of the pioneer biotechnology firm Genentech, uses E. coli bacteria to produce human insulin. 1988, genetic fingerprinting was discovered, which is used today to establish family relationships and also to identify criminals and suspects. 1986, the first genetically engineered plants are grown outside in fields for the first time in the USA. These are genetically altered tobacco plants. 1989, discovery of defective gene for cystic fibrosis by Dr. Lab Chi Tusu at Toronto's Hospital for Sick Children. The year 1990, chymosin, an enzyme that is used in cheese making, becomes one of the first food products in Canada to be manufactured with recombinant techniques. 1997, the world meets Dolly, the ship, the first cloned mammal. UNESCO adopts the Universal Declaration on the Human Genome and Human Rights, recognizing the human genome as a common heritage that must be safeguarded for inappropriate manipulation. 1998, the roundworm becomes the first multicellular organism to have its genome completely sequenced. 1999, German and Swiss scientists develops golden rice fortified with beta-carotene, which stimulates production of vitamin A, thus preventing forms of blindness. The year 2003, the Human Genome Project is being completed, and the researchers at Canada's Michelle Smith Genome Science Centre in British Columbia are the first to sequence the SARS genome. 2005 is the year known for the billionth biotech acre planted for about 8.5 million farmers in one of the 21 countries. The year 2007 is known for the development of first vaccine against human papillomavirus. The first vaccine against human papillomavirus, a cause of cancer, is approved for the use by women and girls in more than 80 countries. The year 2009, Winnipeg's National Microbiology Laboratory completes the first genetic sequencing of H1N1 flu virus, just as the disease is reaching international pandemic proportions. 2009 is also known for a Canadian team of scientists and engineers from the University of Toronto that developed a microchip with nanoscale components to detect chemical markers for cancer, a technique that could make diagnosis much faster. The year 2011, human trials of malaria vaccine are underway and showing the positive results, and this could be the first vaccine against a parasitic infection. The year 2010, first synthetic cell in May 2010, J. Craig Venter Institute created the first fully synthetic self-replicating bacterial cell, which was named Cynthia, while the US government has plugged around $430 million into synthetic biology since 2005. The year 2012, draft genome for wheat. An international team announces a draft of the wheat genome. 2013, the first bionic eyes has seen the light of day in the United States, giving hope to the blind around the world. 2003 also celebrates the 60th anniversary of Watson and Crick's discovery of the double helix. 6000 BCE is the era known for the yogurt and cheese being made with lactic acid producing bacteria by various people. 4000 BCE is known for the Egyptians that baked leavened bread using yeast. 500 BC is known for moldy soybean curds that are being used as an antibiotic. 250 BCE, the Greek practiced crop rotation for maximum soil fertility. 100 CE, Chinese used chrysanthemum as a natural insecticide. The next era is known as the pre-20th century. Let's have a look at the following years that lead to the discovery of many new technologies. The year 1663 is first recorded description of living cells by Robert Hooke. 1677, Antony van Leeuwenhoek discovered and described bacteria and protozoa. 1798, Edward Jenner uses first viral vaccine to inoculate child from smallpox. The year 1802, it is the first recorded use of the word biology. 1824, Henry Dutch-Roquette discovers that tissues are composed of living cells. 1838, proteins were discovered. They were named and recorded by Gerardus Mulder and Jones Berzelius. The year 1862, Louis Pasteur discovered the bacterial origin of fermentation. 1863, Gregor Mendel discovered the laws of inheritance. 1864, Antonin Prandtl invented first centrifuge to separate the cream from the milk. 1869, Friedrich Miescher identified DNA in the sperm of a trout. 1871, Ernest Hope Seller discovered invertase, which is still used for making artificial sweeteners. 1877, Robert Koch develops a technique for staining bacteria for identification. 1878, Walther Fleming discovered chromatin, leading to the discovery of chromosomes. 1881, Louis Pasteur develops vaccines against bacteria that causes cholera and anthrax in chickens. 1885, Louis Pasteur and Emil Brox developed the first rabies vaccine and used it on Joseph Meister. Now let's have a look at the events that took place in the 20th century. 1919, Károly Eréki, a Hungarian agricultural engineer, first used the word biotechnology. 1928, Alexander Fleming notices that a certain mole could stop the duplication of bacteria, leading to the first antibiotic, that is penicillin. 1933, hybrid corn is commercialized. 1942, penicillin is mass-produced in microbes for the first time. 1950, the first synthetic antibiotic is created. 1951, artificial insemination of livestock is accomplished using frozen semen. 1952, L.V. Rauderich and V.M. Lukyanovich published clear image of 50 nanometer diameter tubes made of carbon in the Soviet Journal of Physical Chemistry. 1953, James Watson and Francis Crick describes the structure of DNA. 1958, the term bionics is coined by Jack E. Stelly. 1964, the first commercial myoelectric arm is developed by the Central Prosthetic Research Institute of the USSR. 1972, The DNA composition of chimpanzees and gorilla is discovered to be 99% similar to that of the humans. 1973, Stanley Norman Cohen and Herbert Boyer performs the first successful recombinant DNA experiment using bacterial genes. 1974, scientists invest the first biocement for industrial applications. 1975, methods for producing monoclonal antibodies developed by Kohler and Kaiser Milstein. 1978, North Carolina scientists Clyde Hutschken and Marshall Edgell shows that it is possible to introduce specific mutations at specific sites in a DNA molecule. 1980, the US patent for gene cloning is awarded to Cohen and Boyer. 1982, Humulin Gentech, human insulin drug produced by genetically engineered bacteria for the treatment of diabetes, is the first biotech drug to be approved by Food and Drug Administration. 1983 is the year known for PCR, that is polymerase chain reaction technique, and that is being developed. 1990 is the first federally approved gene therapy treatment that is performed successfully on a young girl who suffered from an immune disorder. 1994, the United States Food and Drug Administration approves the first genetically modified food, the flavor saver tomato. 1997, scientists led by Ian Wilmut from the Roslin Institute reported cloning Dolly the sheep using DNA from 2 adult sheep cells. 1999, the discovery of the gene responsible for developing cystic fibrosis. 2000, completion of a rough draft of the human genome in the Human Genome Project. Now let us have a look at the 21st century events. The year 2001 is known for the Human Genome Project being drafted, and it is being published by the Science and Nature magazine. The year 2002 is known for the rice that became the first crop to have its genome decoded. The year 2003 is for the Human Genome Project being completed, that provided information on the locations and sequence of human genes on all 46 chromosomes. 2008, Japanese astronomers launched the first medical experiment module called Kaibo to be used on the International Space Station. 2009, Kedr's Sinai Heart Institute used modified SAN heart genes to create the first viral pacemakers in guinea pigs, also known as iSense. The year 2012, 31-year-old Zach Water successfully uses a nervous system control bionic leg to climb the Chicago Willis Tower. So this was an insight of the discoveries, the inventions and modifications that provided evidence of the evolution of biotechnology that occurred much before the Common Era.